Just first, I start by thanking the organizers for the kind invitation. And This talk is about evolution, and I'll speak about adaptation in changing environments. Okay, so before I do that, let me first ask what is evolution? Okay, so I'm sure all of you have some idea what evolution is, but let me remind you anyway. So the basic idea is that in a population, there are variations. So for example, we all look different from each other. We have some variation in the nature that we see around us. And this variation is heritable. That is, it can be passed on from one generation to another. And if these variants, they happen to be good in that particular environment, then that variant is selected for. Okay, so that's the basic simple idea. And there are these keywords that I have highlighted: selection, heritable, and variation. So if you understand this much, I mean, that's basically the basic idea about the evolution. However, according to Darwin, we see nothing of these slow changes in progress. We only see that the forms of life are now different from what they formerly were. That is, the Darwin's idea was that, his feeling was that we cannot really see evolution in action. However, now we know numerous examples where we can see evolution happening right in front of our eyes. Here's this classic example that we all know from your school textbooks, that is of the story of this peppered moth. That is, there are these two types of moths, the dark ones, the black one, and the white ones. So in the pre-industrial era, the trees were covered with lichen, and the white the moth was not visible to predators. So the black peppered moth was selected against, and its frequency was rather low. But during industrial revolution, when the trees were covered with soot, it's the black peppered moth which was selected for. And then again in the 1950s, after the cleanup, again it's the black peppered moth which is selected against. So the point that I'm trying to make is that over a span of just 100 years, the frequency of black peppered moth went through an enormous change from a very low frequency to almost frequency 1, again back to low frequencies. So that's one this standard textbook example of rapid adaptation. I'll give you one more example, and this is an experiment on E. coli. And this experiment actually started in 1988, and it's still going on to this day. And the experiment setup is it's quite simple. All one has is a dozen flasks with E. coli, and these E. coli are just given less glucose than they are used to. So now they are in a new environment to which they are maladapted. They are not, adapted to this less food. So what will happen now is, if they want to survive, they have to change their genetic structure. Some mutations have to happen so that this population can adapt to this new condition. In other words, its fitness has to increase if it survives. And below here is shown the experimental result, which relative fitness relative to the ancestor is plotted with respect to time. What again you see here is that there's a dramatic increase in the fitness. In just 5,000 generations, there's a 40% increase in the fitness. Of course, as time goes on, this fitness increase slows down. It's not as fast as it was in the beginning. Okay, but nonetheless, there's something again you can observe in just 5,000 generations, which is not much in the human years. The second point that I want to make from this slide is that there is a real interest and need for quantitative modeling in evolution. So, for example, if you want to understand that why is there suddenly there's the big rise in the fitness and then it slows down, one can sort of come up with a qualitative explanation saying that there's a fitness hill and the population sitting at the bottom of it, and as more beneficial mutations are available at the beginning of evolution, the fitness is low, and as it climbs up, number of beneficial mutations reduces, and near the top of the hill, they're even less, and therefore the fitness is increasing, but not as fast. That's a qualitative explanation. And here, what is shown in green and red are results from a theory, from a detailed theory, and that's the sort of quantitative modeling what one is interested in, and what drew people like me, who are statistical physicists, to this subject. Okay. So what kind of modeling does one do in evolution? So basically, one comes up with sort of two ways of looking at evolution. There are two sort of very broad ways in which theory is used in evolution. So one is about giving a prospective view of evolution. So the idea here is that I have some, two types of individuals, two types of alleles, if you like, the red ones and the black ones, and they had certain frequency. There are some evolutionary forces, which I'll speak about in a minute. Under the action of that, these gene frequencies, these allele frequencies are going to change in time, and then we have a new set, a new frequencies. So this is how a physicist would also think. But basically what we will do is to write down some equations of motion for the frequencies according to how they are evolving in time. This is the time evolution equations. So that's a prospective view, which is to predict the future. There's also a retrospective view of evolution, which is that, I know the frequencies today. There is certain frequency, okay? But I would like to know what are the forces which are responsible for the genetic structure of the population that I see today. What was the direction, what was the magnitude of these evolutionary forces? Of course, we cannot go back in time. What one can do is to make inferences. So that's another way, this retrospective view, which is used using theoretical modeling in evolution. So today I'll stick only to the first one, which is to start basically thinking about some kind of equations of motion and see how things will evolve in future in time. Okay. Okay, so I mentioned that the population is evolving forward in time under the action of some evolutionary forces. What are these forces? So there is natural selection. You already heard about it, and it is mutation. So that's something which all of you have always, have some idea about again. But what is less known is the stochasticity plays an important role in biological evolution. And then there's a fourth category, a broad category of processes called population structure, which is about how is there a structure, special structure, etc. in the population. So it's not really a process, but it's something which you need to add to the model to make it more realistic. But today I will not bother about it. Okay, but then the broad question then is that due to the interplay of these processes, how does the frequency of an allele change with time? So let me say a little bit about each of the first three in brief. Selection. So as I already said that you have some idea about, so here for today, I will just I'll assume that I'll deal with single gene, and a gene has just two variants, which are in this literature called the alleles, the light one and the dark one. And these two come endowed with a quantity called fitness, which for the dark allele I'm denoting by 1 plus s, and for the light one with just it's 1. It's a relative fitness, or the fitness difference is s. This s could be positive, in which case we will call the dark allele to be a beneficial mutation. It could be 0, in which case neutral, or deleterious, in which case s is negative. If it's a beneficial mutation, you expect intuitively that the dark allele's frequency would increase with time. If it's deleterious, in that case, the dark allele's frequency will decrease with time. And if it's neutral, you don't expect anything to happen. Okay, so that's your intuitive expectation. And let me just mention that fitness is not some theoretical construct. It's something which is measured in experiments. For example, in Lenski's experiment, the growth rate is a measure of the fitness. So that's selection. Now, selection has a tendency to, select the fitter one, to localize the population towards the good allele. But that's not all. There's also mutation, which acts just in the opposite manner. So if there's only selection, you have the survival of the fittest. That is, all the population is localized around a fit sequence. But because of the mutations, new variants arise, and that of course is an essential thing for the evolution to occur. So there will be these other variants which perhaps are localized around the fittest sequence. But if you increase the mutation rate further, then perhaps the whole genetic space is filled up. So as mutation rate is increasing, you have these variants increasing with time, increasing with the mutation rate. And actually, one can make these statements more precise. One can actually have phase transitions. And that's something which physicists have worked quite a lot. And I myself got interested in this topic because of this kind of phenomenon. Okay, so we spoke about mutation, we spoke about selection. But now let me come to random genetic drift. So I said that stochasticity plays an important role. And let me illustrate that by this simple example, what I have in mind about what is genetic drift. To put it simply, it's simply a sampling noise. So, but let me explain that. So suppose I have a petri dish which has limited food. So every system has a finite carrying capacity. So there are these, in the parent generation, there are these 5 individuals, the dark two and these 3 light alleles. Now they both reproduce. Let's say there is, each of them produce exactly 2 offspring. However, there's food only for 5 individuals. So somebody has to die. Half of the population has to be removed. One reasonable way to do that is to throw a coin, which is a biased coin, which is biased in favor of the fitter allele. So let's say in this case, darker allele is the fitter one. Then we expect that, when you throw a coin, it's possible that the darker allele gets wiped out more than the lighter one. And you wind up with this new generation in which, although the darker allele was fitter, you're left with a single individual instead of 2 that you started with. So in this manner, because of the constraint that these are finite resources, there is a sampling noise, and that is implemented. That's what basically random genetic drift is. So that's the stochasticity which is there in finite populations when we in finite populations. Now, this stochasticity has some very important effects. It has an important effect. Okay, so I'll come to that in a minute. Okay, so sorry, let me backtrack. Okay, so I told you about mutation, selection, and drift, and I explained to you separately, but one can put them all together in a single model, and that is known as the Wright-Fisher process. So if you open any population genetics book, this is perhaps one of the first things that you will see. So what basically one is doing here is, it's a dynamical process. What we has is that a bunch of individuals, say again the 5 of them, and these 2 steps that I said was selection reproduction, one can sort of eliminate them and implement this process in which each offspring chooses a parent with a probability proportional to the parent's fitness. So this chose this parent and this chose this parent and so on, and you can implement it very easily in your computer. And at some point, a mutation might occur. So for example, here it happened, and then again, these individuals will be produced, which are probability proportional to the parent's fitness. So this can be implemented in your computer. Okay, so this is the sort of the basic processes of evolution that we have in mind, and we would like to understand and see what are the implications. Okay, so now let's imagine a situation in which we have some population of only the lighter alleles, and a mutation happened. Okay, just when the mutation just happens, there is only a very few of those mutants to start with. For simplicity, let's say just say that there's a single mutant. So I have the 5 individuals again, and there's a darker allele which just came in. If it happens to be beneficial, you expect intuitively that it will produce this offspring with more probability, and then if no more mutations happen, then all the population will be of the dark allele type, right? So that's your intuitive expectation. But because this process is a stochastic process, it may happen that dark allele, although it's a beneficial one, it might not produce any offspring. It might happen that the dark allele might get lost. So if nothing else, please take this simple message from home from me, which is that your usual expectation that the beneficial mutation only will win is not really correct. Random genetic drift, because of that, it's possible that the beneficial mutation can get lost, okay, and a deleterious mutation can spread in the population. So that's a simple, basic message to remember. I would also like you to note from the stochastic trajectories that for a beneficial mutation, this stochastic loss is important only at short times when it has very low frequencies. Once it reaches some finite frequency, 5%, 10%, something like that, then it just marches ahead almost deterministically. For deleterious mutant, even if it reaches quite high frequency, it can still get lost. So that's a simple difference between when you have beneficial or deleterious mutant, how the trajectories look like. Okay, so these are the 2 basic questions that these slides motivate. what is the chance that a mutant which is initially rare, that means it's present initially in small numbers, would spread in the population? In this literature, it's called fixation probability. And how much time does it take? That's called a fixation time. So I will not speak about fixation time today. I'll focus only on fixation probability. Now, if you're somebody who doesn't care about evolution, doesn't understand evolution, doesn't matter, let me rephrase this question here. What I'm talking about here is that I have a system with two absorbing boundaries, because I said there are no more mutations possible. So either everyone, if all individuals become the dark type, that's the end of the story. If all individuals become the lighter allele, that's the end of the story. So you have two absorbing boundaries, and one is asking, what is the first passage probability to the specified boundary, and what's first passage time? Okay, so that's the sort of questions that we as a statistical physicist are very familiar with, and we know immediately how to deal with it. So that's the standard procedure of doing that is called the backward Fokker-Planck equation. So forward Fokker-Planck equation, in which you say that, yeah, I start with some initial condition and these trajectories are going to go forward in time, and I want to know how much is the frequency at time t. That's the forward one. The backward Fokker-Planck equation is more appropriate to answer questions dealing with absorbing boundaries. Here, one is fixing the final point. The termination point is fixed, and what is varied is the initial condition. And this probability is always, it's a partial differential equation. So there is a time derivative and two space derivatives. Space here means allele frequency. Here is the drift coefficient, and this is the diffusion coefficient. Both of them are in general dependent on space and time. For the Wright-Fisher process, one can work out that D1, for instance, if I have logistic growth, it's given by x into 1 minus x, the usual standard logistic growth, with a coefficient s. S, remember, I said to you, is the one which characterizes the amount of selection and the type of selection, positive, negative, zero, right? Diffusion coefficient here is coming because of sampling noise. That is the variance of the binomial distribution. So that's why I have x into 1 minus x divided by this, the size of the population. If I have an infinitely large population, there is no noise. Because I have a finite population, there is this drift coefficient, it is a diffusion constant. In any case, we have this PDE, and that's the sort of the standard method of dealing with these problems about fixation probability and fixation time. Okay. What one is actually interested in, I mean, one can of course try to solve the whole differential equation, but one is interested in usually is the eventual probability of fixation, that is to hit one of the boundaries, say x equal to 1 at infinite time. If I have a constant environment, that is selection is not changing with time, in that case, we can put the left-hand side to be 0. And what we have then is a ordinary differential equation. So this describes a time-homogeneous Markov process, and one can basically, do whatever. I mean, you can basically solve it very easily. Today, I'm interested in changing environment. That is, selection is not translationally invariant. I have a time inhomogeneous Markov process. In that case, I cannot put the left-hand side to be 0. And I still have to deal, therefore, with the full PDE. So the simplification that happens for the constant environment does not happen here, and one has to deal with this rather difficult situation. Okay, so that's one way of dealing with it. And that's something which is familiar to physicists. If you're trained as a statistical physicist, you basically know this stuff. Okay. However, there is one simplification. There's an approximation that has been used when I have a beneficial mutant. And since something which is not known to physicists very much, I thought I'll share this simple idea. So the idea is this. So we can, of course, deal with selection, which is, we have a deleterious mutation. The mutant could be deleterious, it could be beneficial, it could be neutral. But if I happen to have only beneficial mutant, if I'm interested only in understanding adaptation, then these questions can be answered within a semi-deterministic approximation. So the idea there is, as I mentioned already, if I have a beneficial mutant, it just has to avoid the stochastic loss when its number is low. Once it's past that stage, it will just march up ahead deterministically. So therefore, one can divide the problem into a stochastic phase and a deterministic phase. Deterministic phase you know how to deal with. You just write down your rate equations. Stochastic phase, well, we should write Fokker-Planck equation, but that Fokker-Planck equation can be simplified. In general, one uses a class of processes called a branching type process, where the important thing is that lineages are assumed to evolve independently. This assumption allows one to make analytical progress. So we have a solution in the stochastic phase, we have a solution in deterministic phase, and then there's a way to splice the two solutions and get the information that one needs. So this is a simple idea, and it's very useful, and there's something which is probably not known much in the physics literature, however. Okay, so fine. So we are interested in evolution in changing environment. So what we did was to use the full Fokker-Planck equation. We also used the semi-deterministic approximation and some numerical simulations to understand that problem. Okay. So before I just wanted to ask, is there any question before I go to the specific model?
Yeah, I have a quick question.
Yes, please.
So, you had shown a branch structure. On the other hand, you had only one variable x. So what does x mean here?
I'm sorry, I should have said x is the mutant frequency.
Description in
I have just two types, the mutant and the wild type, right? So the mutant frequency is x and wild type frequency is 1 minus x.
Okay, so this branch structure does not enter explicitly in the first passage time calculation?
Yeah, you can. Yeah, you can use the. I think branch structure. You mean the branching type process? You can use that. Yeah, you can use that for the first passage time. Yes.
Okay. Okay.
Yeah. Okay. So let me now come to the specific question, specific model. So we are interested in changing environment in general, and today I'll speak only about periodically changing environment. So the motivation being, for example, if in some plants a mutant appears, but maybe it has a growth rate which is different in winter from summer, or when your doctor gives you some medicines, he or she might want to give it to you more initially and then reduce it and maybe increase it again. From the point of view of the pathogen, therefore, selection is changing with time, cyclically perhaps. Okay, so one can of course come up with some sort of this simple model for the selection coefficient, which has several parameters, and one could ask, how does the fixation probability, the chance of survival, depend upon the rate of environmental change? If the environment is changing fast, or slowly, does it depend on that? Well, you expect yes. So how does it depend on it? Does it matter what time the mutant arose? Suppose the mutant arose over here when the selection was going down. Is the chance of survival different when the mutant arose when the selection was getting stronger? Well, you sort of know the answer already, because I said we are dealing with a time inhomogeneous equation. So you expect this kind of initial condition dependence to be there. So we would like to understand, therefore, how does the chance of fixation depend on the frequency as well as the arrival time. Okay. So before I go to any formulas or anything, let me do something simple, qualitative picture. So what I'm showing here you is the mutant frequency as a function of time, and there's several trajectories. And in this limit, I have taken the case in which the environment is changing extremely slowly compared to the time it takes for it to spread in the population. If it's changing very slowly, you expect that basically it's more or less like the static environment, right? Okay. But with static environment, the environment is changing, right? That static environment in which it arose. So arrival time is obviously very important when the environment is changing slowly. Let's take the other limit in which environment is changing extremely fast. So you have rapid oscillations at the level of on time scales of a single generation. So before the population could even track that what is the change, the new generation has appeared. So it will not be able to track these changes in the environment. It will be sensitive only to the average environment. So it will react to the average environment. Okay, so which is different from the small frequency case. However, in this case also, although I said that what is going to matter at large frequencies is the average environment, time still matters. Because you see, this population, when the mutant population, when it's small, it still has to survive at short times in order for anything at later times to happen. So arrival time is still going to be important. Okay, so the words that you're seeing again and again in the last few slides is the time inhomogeneity is important when you are dealing with a changing environment. You have to take care of that. Okay, so let me just show you the answers. So let's try to understand what are these qualitative character. So this is the fixation probability plotted as a function of the frequency of the environmental change. And let's say that the mutant arose when the selection was going down. So because selection is weakening, you expect the chances of fixation also to decrease at small frequencies as well as at the large frequencies. So from the large frequency, this limit from here also it reduces. In other words, there's a minimum in the fixation probability. On the other hand, if the mutant arose when the selection was increasing with time, you expect that the chances of survival would be more, and therefore fixation probability would increase from this side as well as the average environment side. In other words, there will be a maximum in the fixation probability. And one can write down some simple formulas, but let's not worry about that. But the point here is that there is a resonance frequency at which the fixation probability has a minimum or a maximum, and at which and around in the region around which the answers are very different from the static environment case. Now we know again from physics that resonance occurs at the natural frequencies. So what is the natural frequency here? Well, if I have a beneficial mutant, then the natural frequency is simply the growth rate of the population. So if the environment is changing at a rate comparable to the growth rate, we expect answers to be very different. The chances of survival may be very smaller, maybe much smaller, or much larger. Okay, so that's the sort of key point here. Okay, now so far what I described to you, I described to you only dependence on frequency, dependence on the arrival time, but from a biologist point of view, there's one more important factor, which is called dominance. So I'll just very briefly say about that. But let me remind you first, what is dominance? Again, you remember it from your school in the Mendel's experiments. There were these pea plants. When you have round ones and the wrinkled ones, you always see the round pea seeds. So here's another example. If you cross this round seeds corn with a shrunken seeds corn, you see only round seeds. Then you will say that the round seeds corn is a dominant one, or the shrunken seed one is a recessive one. But that's not always true. For example, in the striped dragon flowers, you can have this dark pink and the white ones. These are the two types. But if you cross them, you don't get only dark pink or white ones. You get this light pink. The offspring is very different from the other two. And these things are, so it's not either the type. And one characterizes this by a parameter H, which lies between 0 and 1. Okay, so this is the dominance parameter that one is interested in as a biologist. Okay.
Sorry, Kavita, would you mind wrapping up?
Yeah, I'm sorry. Yeah.
Yeah, would you mind wrapping up? Otherwise, we'll have
I'm almost
left for questions.
Yeah. Yeah, I'm almost done. Yeah. Okay. So there's a standard resultant subject which says that dominant beneficial mutants have a higher fixation probability than the recessives. So what does that mean? At some time in the past, suppose I had a population, okay, and then it acquired a mutation. If that mutation happens to be recessive, it is unlikely that it will spread. But if it happens to be dominant, then it will spread to the population I'll see today. In other words, the dominants are younger. And this actually idea has been used to construct phylogenetic trees of some insects and even butterflies. Okay, this result is, however, valid when the environment is static; it's not changing with time. In changing environment, also we see that for some arrival times, this holds. Haldane's C works. That is, higher the h, higher the fixation probability. But in some arrival times, this idea is reversed. This result is reversed. Recessives have a higher chance of fixation than the dominants. How does that affect these phylogenetic trees? And I don't know what the data on this. So I don't know how this helps. Okay, but so I'm basically done. So what I tried to do was to describe to you a stochastic model of biological evolution. And the reason one of the reasons, I mean, evolution is interesting in its own right, but one of the reasons physicists like me are interested in, because one can use the ideas we have learned from statistical physics to understand and address some questions in evolution. And I looked at a specific model, and I got some, we got some answers, and there's something which we're still looking at. But I think I'm over time as it is, so I'll just stop here. Thank you.
All right, thanks a lot, Kavita. Questions from the audience, please go ahead, unmute yourself.
I have a question, Kavita. This is Debashish.
Yeah. Hi.
From Maiti. Hi. So I was a little lost in the terminology. So when you write the backward equation, there the object that appears is the survival probability directly, which is a function of time. So now, what is the fixation probability actually related to? Because that I see is not a function of time. That's a function of the parameters of the model.
Yeah.
So what is that related to the survival probability actually?
Yeah. So this fixation or survival probability I'm saying is the one that I'm describing here is the eventual one. That at infinite time, I don't care what time, but eventually there is a fixation. Remember, there's two absorbing boundaries here. So it can go to either of them, and I don't care what time it fixes. So I'm just saying that I'm interested in when t goes to infinity.
T goes to infinity, the survival probability.
Yeah.
Okay.
Yeah.
The value of the survival probability at eventually that happens.
Yes. Starting at a value x naught.
Got it.
Yeah?
Thank you.
Okay.
Yeah. Okay. Yeah.
So Kavita, maybe I'll ask you a quick question. So when you mentioned that if the environment is fluctuating very rapidly, let's say on the order of the generation times, I think you said that the cells or the organisms feel the average environment, right? Hello, Kavita, can you hear me? Hi, Kavita. Are you there? Yeah. So I think you said that it feels the average environment, right?
Yeah.
Is that an assumption, or is that something that's actually been tested in an experiment? Hello? Yeah, sorry.
Yeah, sorry, I lost. Yeah, so I think what you're asking is that an assumption or is it something shown?
Yeah, exactly. Has it been?
Yeah, no, it can be shown. This is shown.
I see. And can you tell us a little bit about how this is shown?
You just take omega going to infinity limit here of these equations.
Okay.
Yeah. I mean, intuitively, it's sort of clear, right?
I'm not exactly sure, because often in cancer cells, when you change environments very rapidly, sometimes in certain situations, cells can actually respond almost immediately. Like if I'm adding drugs and then washing the drugs away, sometimes in certain situations, cells can respond pretty instantaneously, and it's not actually feeling the average environment.
Yeah, but it depends. But this is sort of, so I'm not sure, maybe the assumptions is different. But if you have omega to be infinity, basically it's the average, but maybe there's some other assumption which we haven't taken care of.
I see. Interesting. Okay. And are you always thinking of like bacteria or other organisms which are dividing very fast, or this is general?
So I'm actually thinking of populations with a huge recombination, because I'm assuming that only single locus is under selection. Okay,
I see.
so that's a sort of basic assumption here. But if you're thinking of bacteria which are asexuals, so we actually have to do a much tougher job. Here we are dealing with a single site problem. Then we have to look at L sites which are, affecting each other. That's a much harder problem. But that's something which we do intend to take up.
Interesting. Okay. Okay. Any other questions?
Hello, ma'am. I have a question.
Yeah.
So you started the stochastic modeling
Hello?
from the backward Fokker-Planck equation. Hello, hello.
I can't hear you.
So, hello, ma'am. Can you hear me now?
Yeah, it's breaking off, but please go ahead.
Yeah. So you started the stochastic modeling beginning from the backward Fokker-Planck equation. In general formulation, one starts from the generalized Langevin equation, where one defines the kind of potential, the kind of frictional force, the kind of random noise, be it Markovian or non-Markovian in nature. So you just started from the backward Fokker-Planck equation. So is it taking care of any kind of noise or friction present in the system, or
So I am.
there are, of course, there are both kind of present here?
Yeah. So Langevin equation would deal with how the frequency is changing with time. That's a stochastic differential equation.
Okay.
Here, I'm talking about the distribution itself. So Fokker-Planck and Langevin in general are like that. So the Langevin equation will tell you how x naught is changing with time, or rather x is changing with time, the frequency is changing with time. But the probability, if you take the histogram of that, that's what is described as the Fokker-Planck equation. But you can deal with Langevin equation also. But I'm somehow more used to working with the Fokker-Planck.
Okay.
And backward and forward cases
so,
are
and you, yes, of course, ma'am, that I understand. And you try to solve the Fokker-Planck equation. The solution is giving the required probabilities of interest.
That's right. Yeah.
Yeah. And ma'am, one more question I have from that dominant slide, where you have shown that when the two maize species were crossed, the dominant characters were coming in the progeny, right? So, what
That's
is the
right.
probability that in the later generations, recessive characters will also be observed? Because in Darwin's experiments, the first generation was okay, long plants and everything, but later dwarfism was also observed. So, is there any kind of probability which is calculated, or what is that? I would like to know.
Yeah. So that's actually the parameter in the model. It's a parameter H. So in it depends upon the system. So in corn, this trait happens to be either dominant, completely dominant, or completely recessive. So that's a characteristic system. But in other cases, you can have like this pink snapdragon. So it's a parameter which will enter the model to say how much is the probability that you will see the heterozygote to be different from the two homozygotes.
Okay.
So it's not a probability,
So
it's
it is probable,
a
right?
characteristic selection. I mean, I don't know if it's probable, but it's something which is, it says whether it's fully dominant or it's intermediately dominant.
Kavita, right at the beginning, in Lenski's experiments, there were these two red and blue or green curves that you had shown. So can you tell us a little bit more about those two?
So one of them, so I mean, the green curve fits better, of course, but the assumptions, so there are two sort of important things. I'll just say the words, which is the clonal interference and epistasis. So there's the technical words which go in one of the theories. So clonal interference refers to the fact that mutants, when they're arising, they will interfere with each other. Okay, so that's taken care of. And also epistasis refers to interactions between the fitnesses. So these interactions between the mutants and between the fitnesses is taken care of, both of them, in the green curve.
I see.
Okay, and the red one is, so because it's at that time this theory was sort of somewhat newish, and we were trying to understand that if I just take care of one of them or the other one, do I get the right answer? So it's just to show that.
I see. So the message from this slide is that epistasis and clonal interference both are very important to understand the data.
That's right. Yeah.
I see. Very interesting. Okay, if there are no other questions, maybe we can thank Kavita once again.
Yeah, thank you very much.
Yeah, thank you all.
Okay, thank you, Kavita.
Thanks,
Thanks,
Debashish.
Tanveer, and thank you, Sharon, for sharing this session. Thank you all.
All right,
Thank
bye.
you.
Bye, everybody.
Bye-bye.